Anybody come here to listen specifically about fire blight? So, okay. Got one person there? All right, all right. So my name's Ken Johnson, and I've worked on fire blight a long time, and I'm actually going to tell you, I'm going to give you a story of Biocontrol that's about 25 years old of this disease. So, and the reason it's become important in organics, it's always been important in organics, but last year was the antibiotics got sunsetted out of the NLP program, and that was a kind of a, maybe many of you probably read about this or heard about this. But, so you can see fire blight here, and when it gets to this stage, there's not a lot you can do, and so people are pretty frustrated at this point, and especially in young trees, you can kill them real readily with the fire blight bacterium. And so biocontrol is going to play a part in the future without antibiotics. So yeah, I can go. So this is sort of the disease cycle, and I've got antibiotics listed here, and this is sort of where antibiotics would be used. Like, so the disease, the pathogen overwinters in these old cankers in the tree, and some growers will paint copper on those old cankers, and that slows the jump of the bacteria into the flowers. And then once the bacteria gets in the flowers, there's this repeating stage we call the floral epiphytic phase, and that's where biocontrol comes in, and it's really important in that phase. And the pathogen needs to grow itself up to get up to about 1 million cells per flower in order to be able to infect the flower. So this floral epiphytic phase is driven by temperature. So warmer temperatures makes bacteria divide faster, and warmer temperatures encourages bees to fly faster. And so this little circle goes around faster and faster as temperature goes. And when the population then builds to a point, a grower will come in with a sprayer and spray something. And this is where antibiotics were sprayed. And the alternatives are copper, but copper isn't always that fruit safe. That can mark the fruit. It's actually toxic to fruit. And then some other materials, and this would be like your serenades, your bacillus type antimicrobial materials that are considered to be biologicals, but it's really just the things that are made, these peptides that are made in the fermentation that actually lead to control. So I really want to talk about biologicals, where we're going to get something on the flower and we're going to grow something on the flower, and what we learned about this. And in the last talk, we learned about insects attacking insects, right? So, and it's kind of a principle of biocontrol that you always want to go like with like. And I want to get talking about a yeast, so we're going to chase a bacteria with a yeast. And why is that? Well, I want to tell you the story about bacteria chasing bacteria first, all right? So fire blight works like this. So there's the stigma, and that's where the pollen grain lands on the flower, and pollen grain is going to germinate there and grow down the style. Well, it turns out to be like a little petri dish in nature. It's nutrient rich, it's hydrated, everything's just right. So a pollen grain grows there beautifully, but so does a fire blight bacterium. And the fire blight bacterium will grow itself up to about 1 million cells there, and then it actually then has to slide down the style with a little bit of moisture, ends up down in the floral cup, and there's little openings in the floral cup where the nectar comes out, and that's where the pathogen goes into the flower, and that's what causes the disease. So it's kind of a two-stage thing, unusual for a pathogen, but the pathogen grows there, slides down the style, and then infects the flower down there. And what's interesting about this is that, well, this stigma will basically grow anything. So you can put anything on the stigma and it'll grow there. And that's how you get to biological control. So we've been studying this for a long time and trying to figure out why it works and why it doesn't work, etc. So just an example of this floral epiphytic phase. So pear bloom, apple bloom, temperature line. So this is a cold spring during pear bloom, then a warm spring during apple bloom, and sort of vice versa, warm pear bloom and cold apple bloom. And these little dots are our measurement of a bacterial biocontrol agent on trees we didn't treat. All right. So we sprayed some trees in the orchard, then we went to the trees we didn't treat, and we say, did the flower get colonized by this bacterial biocontrol agent? And you can see that when it's cold, it doesn't happen, and when it's warm, it does happen, and vice versa. It doesn't matter if it's pears or apples. And that's this little floral epiphytic phase turning around. The bees are coming to the flower. They're picking up whatever's there. They move the microbe to the next flower, and so on. So when it's warm, the biocontrol agent spreads, the pathogen's doing the same thing, and when it's cold, fire blight doesn't usually happen, right? So it's very much a temperature-driven disease.
What was the bacterial and biocontrol agent?
This bacterial biocontrol agent is going to come up in a second, and in this case, it's Pentoia glomerans that used to be called Erwinia herbicula. Yeah. So let's talk about technology. So this is a close-up of stigmas. The stigma has all kinds of little nooks and crannies in it where microbes like to grow. And so this is a complicated slide. Right down in the corner here is the first thing that proves that I was working on this 25 years ago, right? This got on a magazine cover for ARS, actually, that we were working on biocontrol. So in the purple here, this is biological technologies, and I've got a whole lot of technologies here. I'm not going to go into them all, but these are all bacterial biocontrol technologies, okay? And one of these is now called Blight Ban A506. It's a Pseudomonas fluorescens that came out of Steve Lindau's lab. And then Blight Ban C91 was one that we got commercialized, but it was actually first selected in Michigan by Carol Ishimura. If anybody knows who she is, she's at the University of Minnesota now. And that was another one that we selected. And these are just some comparisons. This is what oxytetracycline did for us in inoculated field trials, sort of a so-so product. This is what streptomycin does, and this is what many organic orchards, especially in the eastern half of the United States, didn't want to lose their streptomycin because it was so effective. And then there's something we call integrated control, which starts with biological control and then finished with an antibiotic. And this has become an important concept for us, which you'll see in a second. But so like this treatment here would have been biocontrol first, then oxytetracycline second, and that gives reasonably good control. And I just wanted to make a few points about this. You see how biocontrol kind of leveled off, you know, and so these numbers on the bottom here, this is a mixture of Blight Ban C91 and Blight Ban A506. That's 23 field trials we did, and we got an average of, saying the water control is 100, we got an average of about 50% control, doing that 23 times, all right? So we did that 23 field trials from 1991 to 2008, and we got an average of about 50% control. And biocontrol is sort of leveling off there, and that's what got to be. After you do this long enough, you keep doing the same thing long enough, you start to wonder about these kinds of things. So just an example of one of these technologies that we worked on to improve biocontrol. If we added iron to the flower environment, we switched the one A506 bacterium from just being a competitor, sort of a microbial weed, to actually producing its own antibiotic, just from the presence of iron. And you can see how this kind of works in some trials. Water control gives you 300 fire blight strikes, and the Blight Ban actually gave you more fire blight strikes, but we put Blight Ban with the iron, and now we're down to 100 strikes. That's your antibiotic, and then we do this integrated control, A506 plus antibiotic in the second treatment, and now we're down to 50 strikes. So that was one thing we worked on, was putting iron in, and we have some growers that still do this in the Hood River Valley in Oregon. So I circled here, this was Blight Ban averaging about 30-35% control. And then this is where we add the iron in. That's just Blight Ban with iron, and we improve control somewhat, right? But the question we got interested in was, why can't we get better, right? So I'm more interested now in this purple line here, and why couldn't we get like down to where streptomycin is, right? And that became a big question for me in trying to understand biocontrol. And so I actually went to the literature, and I found every study where someone had done a dose of a biocontrol agent with it and then measured the response, all right? So just dose-response relationships in biocontrol. And nobody ever used the same way of summarizing the data. So I went ahead and I made my own way of summarizing the data, which was just to put the biocontrol density on the x-axis and what they got in terms of disease control on the y-axis. And I kind of got the same pattern that just kept showing up, was that it just took a little bit of the biocontrol to get a pretty big response. and then you could keep adding biocontrol to the system, and it took a lot more biocontrol to get any more response. And then it sort of seemed like the person doing the work quit, and that there was always, I could always draw a red line that said, that's as far as I could get biocontrol to go, right? And so I called this the asymptotic limit to biocontrol. And what's represented by this little red bar here is the amount of disease control that we're not achieving by using a biocontrol agent, right? Does that make sense? Okay. And it's more interesting down here is like, here's the same system, except the pots of plants or whatever they used were side by side, but this was a good biocontrol agent. It's becoming asymptotic at closer to the x-axis, and this was a not so good biocontrol agent. It's becoming asymptotic at a higher level. So the size of the bar here is fairly significant, right? So the point here is that biocontrol dose-response relationships typically show an asymptotic limit to control, and this is a frustration of biocontrol. So I go back and look at my fire blight slide, and I say, well, I've got this purple line here. I'm putting on high doses of biocontrol agent, and they're growing on the flowers, etc. I'm pretty much out there at the limit of dose I can apply to the tree, and I'm getting an asymptotic limit of about 60% control from biocontrol, and I don't understand it because this stigma is perfectly supportive of growth of bacteria, of biocontrol bacteria, and of the pathogen. So got interested in this question is asymptote equals 0.6, 60% controls our upper limit of control, and why, all right? So that got to be something curious to me. And so I decided, like, okay, how can we answer this question? And I said, well, what would be the perfect competitor? Well, the perfect competitor would be the pathogen itself, except the pathogen that can't cause disease, right? So we did a little bit of genetic manipulation with the pathogen, and we did a knockout of that gene right there. And that gene right there is the master switch to making a pathogen, making it pathogenic. So with any pathogenic bacteria, there's a series of genes in there that turn on when it wants to be a pathogen, but are turned off when it wants to not be a pathogen. And we said, well, this is about growth on stigmas. It's not about being a pathogen. It's a place where it needs to grow up its population. Then when it gets down into the plant, it can turn on its genes to be a pathogen. And then, but if we make a competitor that doesn't have that ability, we should have a perfect competitor, right? That was our idea. And did it work? Well, to make a long story short, the asymptote was still 0.6 or 60% control, and with these guys too. And so the question then was why? That was what we were trying to figure out. So did a series of experiments with putting bacteria on stigmas. So we're just talking about putting bacteria on stigmas. And I said, okay, what's the epiphytic yield? How many bacteria, how many angels can I get to dance on the heads of these pins, right? How many bacteria can I grow? And so the number here, that's about 1 million. That's 1.5 million over there. I'm closer to 9 million, I believe, and there I'm about 1.5 million. That's how many wild type cells I could grow on a stigma, right? So then I asked the question with my mutant strain, which in a test tube will grow the same way as the pathogen does. I can't tell a difference in the growth rates in a test tube, but I put the pathogen now, or the mutant on a flower tip, and what happens is I get a whole lot less of them, right? So now I'm only growing this many mutants relative to that many wild type pathogen strains, or this many mutants relative to that many. And they thought, well, that's really interesting. And then they did the other thing was to mix a tiny bit of pathogen in with the mutant strain. And what happens is that you can grow all kinds of mutant strain on a stigma if you have a little bit of pathogen in the mix, and way more mutant strain if the pathogen's there than if you just put the mutant strain on by itself. All right. So that's a kind of a curious result, right? And so we decided that this result, and we did a whole bunch of other things too I'm not going to tell you about, but what's going on here with this pathogen is that it's making some kind of like a micro disease on the stigma. So the stigma's there, there's a little tiny bit of micro disease on there, somehow the pathogen is altering the stigma's habitat to make it more conducive to growing other bacteria on the stigma, right? So the pathogens there, you can grow this many. You put in a little bit of pathogen with the avirulent strain, and you can grow a whole lot more avirulent strain than if you just have the avirulent strain itself. So something about the pathogen being there modifies the habitat to be able to grow a lot of bacteria there. So that was sort of an interesting observation, and we concluded that, well, the answer is why this asymptote to fire blight control? Well, the best answer we've ever been able to come up with is that the pathogens have special abilities relative to the non-pathogens, okay? So, that seems like a frustrating answer, doesn't it? All right, so here we are. What about the yeast? All right, so you already know what the answer is. I'm going to tell you that the yeast has a special ability to grow on flowers relative to the pathogen, whereas in the last slide, the pathogen had a special ability to grow on the flowers relative to the mutant. Now that I'm going to tell you that the yeast has a special ability to grow on the flowers relative to the bacterium, okay? And that's how we get back to a yeast controlling a bacterium. So back to integrated control, and I said there in the beginning, if I put on a biocontrol agent and then I put on, say, an antibiotic after, I get better control. And sort of by deduction, we figured out, well, the biocontrol agent is working on the stigmas, and that means that the antibiotic is probably working down in this floral cup where the infection occurs, right? And so we've used this concept a lot now to really ask questions about products for biological control and what really works. And so think about it at the yeast now. What do you think the yeast does? Well, the yeast likes to grow there, right? So why doesn't a bacteria grow there?
Aerobic conditions or something like that?
It's not anaerobic.
No sunlight.
No, sunlight had nothing to do with it.
Nutrients.
What's a bee looking for if it goes down into that part of the flower? Nectar. What about nectar?
Sweet.
Sugary. Okay, what about, okay, so if you got a jar of jelly, what do you see growing on the top of it?
Mold.
Molds. Do you ever see bacteria growing there?
Not too much.
Not too much, and that's exactly the reason, all right? So here we have our special ability of the yeast, right? The yeast can grow there. It loves to grow there. Bacteria don't like to grow there. And so, and if I knew more, I'd tell you more. But that's about what I know, right, at this point, all right? So I'll show you some pictures here, okay? So this is a picture of that surface of the floral cup in a pear flower, which is very open. And you put the yeast in there, and you can even see some of these yeast in here are actually in the process of dividing. You get essentially a biofilm of yeast on this floral cup. Those big things there, those are pollen grains, right? So you can see how small these yeast cells are. Bacteria don't like to grow there. So for some reason, this biofilm of yeast on this surface is doing a really great job of controlling fire blight. And we don't really understand completely why, other than they're there, right? So how do we know they're there? Well, we just do experiments like where we wash and measure the populations on stigmas. So this thing right here is a Pentoya strain that we sprayed there, and we look at how many stigmas are colonized, and we've got 60 to 70% of the stigmas colonized. This is yeast. Yeast is I sprayed yeast twice here. Yeast is colonizing 100% of the stigmas. So the yeast grows on the stigmas too. Same thing in this experiment. But we look down in the floral cup and we find how many flowers have yeast on them. Well, in the floral cup, all of them have yeast on them. We look how many of them have, where we can actually measure a bacterial population. These are actually really tiny populations. Well, we can hardly find the bacteria in the floral cup. So that's why this is such an interesting thing. It's sort of a different game completely in fire blight biocontrol. And the yeast also spreads around, so if we put the yeast on at 20% bloom, almost all the flowers get colonized, even the non-treated flowers get colonized. So it's spreading around out there. And here's a trial we did last year just to see if we can control fire blight with yeast. So water treatment here. We're getting about 12 fire blight strikes a tree. Streptomycin, we're getting 1 fire blight strike a tree. Oxytet, about 1. This is Serenade, which is one of those antimicrobial peptides. We're getting about 6 strikes per tree. And then this is the yeast, and the yeast actually was the best treatment, though this is kind of statistical noise here. But all of these get the yeast here, so you're averaging between 1 and 2 strikes per tree. Yeah, just about done. So you can see how effective it is. If we were talking again, remember about my bacteria, we'd be out here somewhere. So I'm going to leave it there. I'm going to come to conclusions. So fire blight is amenable to biocontrol. Biocontrol bacteria, however, are restricted to stigmas and compared to the pathogen or poor competitors on this surface, and this creates a limit to control, which was surprising to us and something that took us a long time to figure out. This Basidium pullens is a black yeast. It grows on stigmas, but importantly, it also grows in the floral cup, which apparently shifts the asymptotic level of control. Mechanisms for this asymptotic shift by the yeast are not well understood, other than it grows in the floral cup. Is it nutrient competition? I don't know. Is it induced resistance? Say the presence of the yeast is somehow turning on defense genes inside the flower. I don't know. That's a possibility. And importantly, there's a product now that's made of this Basidium pullens, and it's actually a very aggressive yeast. It actually can be a pathogen itself under certain circumstances. So what I'm suggesting to people that are interested in biological control of fire blight is that they look at yeasts for control of fire blight and not to continue to look at bacteria. All right?
Since we started a little late, I think we can do 2 quick questions now, and then we'll move on to Rick's presentation. So,
Oh, I had 2 questions.
All right, Rick.
everybody else, write yours down. We'll get
Did you
to them
do
at the end.
artificial inoculations?
Yeah, these are all inoculated trial. We missed the pathogen on these trees at full bloom.
So in those early, the early testing that you were doing the trials, it's possible that it was an overload.
It's easy to overload the system. Yeah, and I do think that any biocontrol that you see here works better in a grower's orchard than it does in an inoculated trial. Absolutely. Though growers will tell you, because it's a bigger test than your test, there's so many conditions out there and so many orchards than just my orchard that growers will kind of run the spectrum of experience with biocontrols that work only partially.
The other question I'll just save for the end.
All right. Question?
Are there differences in the varieties with the effectiveness of the yeast?
There's not so much difference in the ability. No, I don't really think so. In terms of the ability to grow microorganisms on flowers, I've never really. We look at pears and apples just almost the same. There are differences in the susceptibility of the cultivars to fire blight. But even like, say, a cultivar like Red Delicious, which is quite resistant, and the old days, like in Washington State, they grew a lot of Red Delicious. They didn't have so much fire blight, but now they're getting rid of Red Delicious. You can still get an infection on Red Delicious, and it just doesn't run very far. It's a real short little thing, and the flower spur might get killed, but you don't get the major damage that you saw like in the first slide there. And with the newer varieties, in Washington State now, 20,000 acres of organics, apples and pears, mostly apples, and almost all Galas and Fujis, which are very sustainable.
Great.
So we can ask another question at the end there.
All right. Thank you.
Yep.